Okay, it is 12 p.m. on the dot, so we'll go ahead and get going. Welcome everybody to our Plant Phenomics Fridays seminar series. I am kind of subbing in today for Carolyn Lawrence Still. I'm Dior Kelly, I'm assistant professor here at Iowa State. So before we get started, I'd like to thank our funding and sponsorship for this program. It's been a great seminar series supported by USDA NIFA, the HIPS program, High Intensity Phenotyping Sites, a multi-scale multimodal sensing and sense-making cyber ecosystem for genomes to fields, as well as the AI Institute for Resilient Agriculture. And I'm glad all of that was actually on a slide because there's no way I could remember that detailed information. So for today, we've got two awesome speakers, Anjali Iyer-Pascuzzi from Purdue University and Koushik from Iowa State University. So I'll first begin by introducing Anjali and then hand it over to her. So she grew up in LA. She got her BS degree in molecular environmental biology from UC Berkeley. She then went to Cornell to do her master's work in plant path with Greg Martin, and then got her PhD in plant genetics with Susan McCouch. She then went on and postdoced with Philip Benfey at Duke, studying root biology. So we have a shared love of roots, and then started her own lab as an independent researcher at Purdue in 2013. She combines her love of plant path and root biology in her research program, which is what she's going to talk to us today about. And a major goal in her lab is to identify the molecular mechanisms underlying root-mediated resistance to soil-borne pathogens. She uses this cool system of tomato with Ralstonia as a really nice pathosystem to use as both a model and kind of spanning different sort of biological scales within this system. So from root cell types, so very cell-specific types of responses, to whole roots, more kind of organ level biology, and combining tools from cell and developmental biology, genomics, phenomics, and quantitative genetics to discover mechanisms of resistance. And outside of work and research and teaching and all that good stuff, she's an avid runner and a mom to 3 crazy boys, 2 of which who are twins. And I'm going to hand it over now.
Great. Thank you, Dior, for that introduction. And thank you for the opportunity to speak today and tell you about some of the work that we've been doing around phenotyping and disease. I was originally going to talk both about root and shoot phenotyping. And in fact, roots is most of what we do, except this particular project is about above ground phenotyping. And that's really all I have time today to discuss. If you want to talk about roots, though, at any time, please do, maybe afterwards in the questions. So most of the work that I'll be talking about today, hold on, let me find my pointer, there it is, has been spearheaded in my lab by my postdoc, Valerian Moline. So my lab focuses on understanding mechanisms of disease resistance in plant roots to soil-borne pathogens. And one of our major questions is what genes and genomic regions provide resistance to different pathogens. And so we think about phenotyping a lot, and we use phenotyping in just about every type of question that we have in the lab. So today I'm going to tell you about how some above ground phenotyping has provided insight into disease resistance. So the major disease that we study is called bacterial wilt, and it is caused by a soil-borne plant pathogenic bacterium called Ralstonia solanacearum. Ralstonia has a very wide host range. It infects over 200 plant species, and it is found throughout the world. It thrives in really hot, humid regions, like you can see here. And it's a very diverse pathogen. So it's actually 3 different species that are distributed worldwide. In my lab, we focus on a strain of Ralstonia called K60 that is found in the southeastern United States, really common in Florida and Virginia. So again, it's a root invading pathogen, lives in the soil. First point of contact is the roots. And it often gets into root systems through wounds that are created when lateral roots emerge from the primary root. And if you can imagine it as this red oval here, it enters into the root system and it makes its way to the xylem. It has to colonize the xylem or it cannot cause disease. And so this is a scanning electron micrograph of bacteria in a susceptible root in a xylem element. Once in the xylem, it moves from the root to the shoot, and it secretes an exopolysaccharide, which is essentially like a slime. And this plugs up the xylem and it prevents water movement from the root to the shoot. And so what you get is the above ground wilting phenotype shown here in a susceptible plant. And here's a resistant plant. So resistance in tomato, as in other crops, to Ralstonia is quantitative. And this means that it's caused by many genes, each of which have a small to moderate effect on overall resistance. So you see a lot of partial resistance. You also see a lot of intermediate phenotypes, plants which are partially wilting. The major source of quantitative resistance that we use is from a genotype called Hawaii 7996, shown here. And you'll also hear me talk a lot today about this genotype, West Virginia 700. We have some idea of resistance mechanisms in Hawaii and in other resistant tomatoes. We know that resistant plants do get infected with bacteria. So here you're looking at light micrographs of tomato root cross sections. Here is a resistant line, Hawaii 7996, and susceptible West Virginia at 0 hours post-inoculation and at 72 hours post-inoculation. And these red stars indicate where bacteria are within the xylem. And so the point here is that in the resistant plant, bacteria are restricted where they are in the xylem, whereas in the susceptible plant, they move out and they're found throughout the xylem. You can see that over here as well. Here's our resistant plant. These are scanning electron micrographs, and here's our susceptible cross sections of roots. And one thing to notice is that in the susceptible plant, you get this biofilm that forms along one of these big TE elements here. We don't see this happening in the resistant plants. So we also know that resistant plants are able to restrict movement of bacteria from the root to the shoot along the xylem. And we know that these plants can suppress genes that are involved in auxin pathways. But even though we have some idea of the mechanisms that are important for resistance, we still don't have a really clear idea of what genes and genomic regions are responsible for resistance to Ralstonia. So this is our major question. And we approach this in many different ways in the lab, but the one I'm going to talk about today is a QTL analysis to identify genomic regions that are involved in tomato resistance to the US strain of the pathogen, and that's often called K60. Okay. So just real quick, remember principle of QTL analysis, we need both phenotypic information and genomic information, and we need a large population to phenotype. And so we phenotyped a recombinant inbred line population that was derived from our resistant parent, Hawaii 7996, and our susceptible parent, and then taken by single seed descent to F9s, and we phenotyped the F9s. So wilting can be really easy to phenotype. Traditionally, it's phenotyped on a scale of 0 to 4, when you look at the percentage of leaves on the plant that have wilted, where 0 is equal to no wilting, your plant's 100% healthy, and then 1 through 4 are increments of 25%, where 4 is a plant that is 76 to 100% wilting, pretty much dead or on its way to dying. And so if you give people images, this is pretty clear on the ends of the spectrum, right? I think just about everybody would agree that this is a 0, and just about everybody would agree that this plant is a 4. But it becomes much more challenging to phenotype when you consider the intermediate parts of the spectrum. And again, this particular disease has a lot of partial resistance. Partial resistance is manifested as this partial wilting phenotype. It's also highly influenced by the environment. So depending on how much humidity you have, you can have different levels of wilting. So if you give people a plant that looks like this, some people might give you a 2, some people might give you a 3. What's the difference between 40% wilting and 55% wilting? If you give people this plant, you might get a 3 or a 4. And this is where it becomes challenging when you're doing a QTL analysis, right? Because you need to be able to accurately phenotype if you're going to identify QTL. Okay, so this was the problem, and we decided to approach it using digital phenotyping methods. So we first designed a really low-cost phenotyping system. So I'm sure many of you have seen this before. This is a commercially available turntable. It turns a user-specified number of degrees, stops, the camera takes an image, and you can see that here, and you can go 360 degrees around the plant. So we use this to take 8 images per plant, one every 45 degrees. And we did 5 biological reps of our population. So with controls that ended up being about 1,000 plants. And we looked at 5 time points across disease progression from the day before inoculation, which we call day -1, to 6 days post-inoculation when our susceptible parent has completely wilted. You will notice that this is in our very super fancy basement storage room. We still get really good images. We do at Purdue have an excellent phenotyping facility, but at this time they were just starting up and they really weren't too thrilled about us putting a pathogen into their facility. So we decided to go this route. Okay, so the next thing we needed some help. We needed to collaborate. And so we went to some engineers on campus who are image analysis experts, and we've been collaborating with Dr. Ed Delb's group over in the College of Engineering. And we've been working with them to think about what is wilting? How do you categorize it? How do you characterize wilting from these images? And so we came up with 3 different categories of wilting descriptors. The first one that I'm showing here is based on color analysis. So here is our susceptible plant that's been inoculated. So again, from the day before inoculation, -1 DPI, through to 6 days post-inoculation. And this is our susceptible plant. This is the parent that has been mock inoculated, okay? And so what we're looking at here is the histogram of the A channel in the LAB color space. LAB color space is just another type of color space, like RGB, it's one type. And so the point here is that at -1 DPI, this is the blue peak here, you're here, and then this shifts as you move from -1 to 2 to 3 to 6 days post-inoculation. Okay, so it's this shift that we're interested in. This doesn't happen when you look at the mock plant. Everybody pretty much lines up at the same point. So we thought, okay, we'll use this shift as one color trait. Then we looked at convex hull analysis. So this is a very typical way of measuring shape. And here you have -1 DPI in a susceptible parent through 6 DPI. And we looked at the width, the area, the height, and the perimeter of the convex hull and how that changes over time. And then the third category had to do with the center of gravity analysis. And so for this, we took our images and we found the plant mass. And then we divided the image into 2 along the stem, okay? And then on each side of the image, we found the center of mass. And so that's denoted here by this red square. And for one descriptor, we called it the center of mass X distance. It's the distance between these 2 points on the left and right image. And then we also use the center of mass height, the distance between the center of mass and the bottom of the stem. And so when you do this in the resistant and the susceptible parents, you can see how this changes in a susceptible plant as it's wilting. The center of mass X distance should get smaller. And indeed, when you quantify that, it does. So here's our resistant parent, our susceptible parent, and our recombinant in red lines, days post-inoculation. And you can see that this doesn't really change in the resistant plant, but it gets significantly smaller in the susceptible plant as it wilts. So then we had our descriptors. We wanted to make sure that they were able to differentiate resistant and susceptible parents in our experiments. Okay, so shown here, this for each of these boxes is one of the traits that we were looking at, and these values have been normalized across experiments. And in this gray on the left is the resistant parent, in the middle is the susceptible parent, and the green boxes on the right, this is the recombinant in red lines. And each of these columns is a separate experiment. And so what you can see is that for most, but not all, for most of these descriptors, they are able to separate resistant parents from susceptible parents, the resistant parent from the susceptible, with the exception here of the color-based metric. So here you can see it really can't differentiate the resistant parent from the susceptible, and so we decided not to use it further. So once we had descriptors or metrics that we liked to describe wilting, then Ed's group built a pipeline where they took our original images, they color corrected them based on this fiducial marker down here and found the plant mass, and then analyzed the descriptors from the mass. So in addition to our digital phenotyping, we also had what we call expert-based phenotypic data. And this is just a fancy way of saying that a graduate student went in and quantified the percentage of leaf wilting daily on the populations. We had this data, the human eye-based data, for 2 years, for 2016 and for 2019, but we only have the phenotypic data for 2019. Then remember, we also need some genotypic data, so we use genotyping by sequencing along with Aaron Sparks at the University of Delaware to generate a new genetic map. And then we did our QTL analysis. And so Valerian identified 20 QTL in what we call 10 QTL clusters across the genome at 6 days post-inoculation. So there is a lot here. You don't need to read all of this. Here we've got our chromosome. Okay, there are 12 in tomato. This is our QTL cluster. This is defined. BWR stands for bacterial wilt resistance. The 2 has to do with the chromosome that it's on. The 1 is whether it's the number of QTL on the chromosome. Our position, the left marker, the right marker, and the trait name. So we defined a cluster as QTL that had the same left marker and the same right marker, but different traits, okay? So within these 10 clusters, we had 3 types. The first type, shown here on chromosome 3, were QTL that we identified both by our expert-based visual notation, so both by eye, but also with our image-based descriptors, okay? So you can see here, this cluster BWR 3.2, we identified it in 2016 by wilting, and we also captured it in 2019 with our image-based metrics. So this QTL appears to have something to do with area, okay? We only had one where we identified it both ways. Most of our QTL fall into the second type of cluster. These are QTL that were identified only by our image-based descriptors. Okay, so you can see, for example, on chromosome 10 here, this particular QTL that was picked up by traits that both quantify plant width. And then our last group were QTL that were identified only by expert-based visual notation. So this was only one cluster, and we found it both by eye in 2016 and in 2019. We did not identify this particular QTL with any of our image-based descriptors. And we are not sure why, but we do have some ideas. I'll talk about that in a little bit. Okay, so just to dig a little bit deeper, here is the QTL on chromosome 3. And here's our wilting score in 2016. And you can see that it has the same peak as 2QTL for area. We are particularly excited about this QTL because there are a number of different defense-related genes under this peak, although there's still a lot of genes underneath this peak, including leucine-rich repeat receptor-like kinases. So then QTL on chromosome 10, remember this group is only by image-based descriptors. And so here we have one for width. And then the QTL on chromosome 6 that was only identified by expert-based visual notation here. Okay, so that's great, but how do these QTL compare to those of other studies in tomato, to different strains of Ralstonia? So ours was the first time that anyone had looked for genomic regions that were important for the US strain, but there's lots of other studies out there that have done this using the same population to other strains of Ralstonia. And those studies have identified 2 hotspots within the genome, meaning there's a lot of QTL, or there are QTL that continuously pop up at those spots. And the first one happens to be on chromosome 6, where we also identified a QTL by eye. So these are simply other studies that have found QTL in this region to different strains of Ralstonia, shown here, and ours would pop up down here. All of these other studies also used visual notation to identify, to phenotype. But then there's this second hotspot that other studies have found on chromosome 12, okay? And we also identified a QTL here on chromosome 12, right up here. And this one we only detected with image-based phenotyping. We did not detect it using visual phenotyping. All of these other studies did use the visual phenotyping. The rest of the QTL, those on chromosomes 2, 3, 4, 5, and 10, these all appear to be novel, meaning they haven't been detected before for resistance to Ralstonia. So one of the questions that we were particularly interested in was whether image-based phenotyping can identify QTL earlier than by the human eye. And so, yes, it can. So again, there's a lot here. If you look down here first, this is what we were just looking at. Remember that we had taken images at 3, 4, 5, and 6 days post-inoculation, also the day before. And so we ran our QTL analysis on each of those days. And here is BWR 3.2, that's the 2 stars. This is the one down here that has both our visually assessed wilting score from 2019 and our image-based scores. And what you can see if you look back at 5 days post-inoculation, at 4 and at 3, we are picking up QTL 3.2 much earlier than we can see it by eye. So we cannot capture it visually at 3 days post-inoculation, but we can capture it with our digital descriptors. Something is changing that has to do with width. Okay, so to conclude then, non-destructive 2D digital RGB phenotyping, sort of like old-fashioned phenotyping, really is how I think about it, can identify novel and previously identified QTL for disease resistance, can detect QTL earlier than by the human eye. We did, though, miss one cluster, right? The one on chromosome 6, that hotspot. Maybe if we had identified it with another type of imaging, we would have picked it up. But I think it might have to do with the fact that our images are all in 2D. So we took 8 images all the way around and we averaged those, but that's not enough to give you 3D that you're capturing with your eye. So maybe if we went back and did it in 3D, we would capture that. So then I want to acknowledge my postdoc, Valerian Moline, who spearheaded this work, Denise Caldwell, who did a lot of the phenotyping and the preparation, Ed's group, Sriram Baireddy and Chengyi Yang, 2 graduate students in his group. And this project was funded through a seed grant at Purdue between the College of Agriculture and the College of Engineering. And with that, I will take any questions.
Thank you, Anjali. That was excellent. So I think we're going to move to the second speaker and then do questions for both speakers at the end, if that works for everyone. Koushik is currently a 5th-year grad student here in the electrical engineering grad program at Iowa State University. He is jointly supervised by Bhaskar Nagasubramanian and Sumik Sarkar. His research focuses on deep learning applications in plant phenotyping. He was a visiting student at University of Tokyo Field Phenomics Research Lab and at a data science intern at Corteva in 2019. Prior to joining grad school, he worked on satellite remote sensing applications in sugarcane in Bangalore.
Hello, everyone. I'm Koushik. Today, I'll talk about self-supervised learning for image-based plant phenotyping. My advisors are Bhaskar and Soumik, and my collaborators are Sara, Ashish, Arthi, Soumik, and Bhaskar for this project. So deep learning models are a part of broader family of machine learning methods based on artificial neural networks. Deep learning generally achieve high accuracy with large label datasets and massive computation. The goal of supervised learning is to learn visual representations from label data. Data labeling can be difficult and expensive. I think many previous speakers have told this, Colleen, Arthi, Bhaskar, it sucks, and especially for graduate students and undergrads. The ImageNet dataset, commonly used by computer vision community, has 14 million images and 22,000 categories, taking 22 human years to label. In this image, you are seeing potassium deficiency of soybean disease being classified into one of the 9 classes. To train this deep learning model, label dataset was used. And manual annotation for plant domain is especially challenging because we need domain experts. So next, we are going to talk about transfer learning. In transfer learning, the model weights learned for one dataset can be used for another application. The image on the left shows a model trained for soybean disease classification being transferred to a mung bean dataset. So what's the advantage of this? We need not have large amount of data in both the domains. Using a large amount of data set in one domain, we can transfer the model for the another domain. So, but here, an important point is for the pre-training of the model, we need label data. So first, we need large amount of label data for soybean diseases, and then we train a model, and then we classify it, classify the diseases, and use the pre-trained encoder for the soybean data for the fine tuning to the mung bean data. And transfer learning has been commonly applied in deep learning community for a long time. Now, but we are going to talk about another way of transfer learning where for the pre-training, we are not going to use labeled data, but unlabeled data. So the goal of self-supervised learning is to learn general visual representations from unlabeled data. The learned model or image representation obtained from self-supervised learning can be used for many downstream applications. So in this image, you are seeing, we are going to artificially create labels by rotating our input images. So we are not asking for labels from any grad students, but what we are going to do is, we given an image, we rotate it into 4 different angles, and then try to predict the rotation of the image using a neural network. So here, since we are not asking for any label data for a pre-training stage, we can use huge amount of unlabeled data without any challenges because we have removed the label bottleneck. And on the left, we are seeing an image of a bird being rotated into 4 different angles. And for each image, we do the rotation into 4 different angles. Imagine we have 10,000 images. The goal of the deep learning model is to predict the classification of the rotation angle from the training data. So once we have this trained model, we can use this trained model for subsequent application. So again, to summarize, in self-supervised learning, we use unlabeled data for the pre-training stage. And once we do the pre-training using unlabeled data, we transfer it for downstream applications. So here the workflow is, first, we rotate our images into 4 different angles, for example, and then train a model. Use the model weights as initialization for the new task, and then fine-tune the representation from the pre-trained model for transfer learning to the new task. So one of the common approaches for self-supervised learning is contrastive learning and simpler. Simpler is a simple framework for contrastive learning of visual representation. An image and its augmented version are passed to the model, and we need for both the image and its augmented version the representation to be similar. What do we mean here? So given an image, we randomly do data augmentation into different illumination conditions, cropping, color jittering, and then flipping. And for all these data augmentations, we want the representation of the deep learning model to be invariant to the data augmentation done in the pre-training stage. So once we train our model using this maximization similarity loss function, we can use the representation for the contrast learning method for a downstream task. So to explain it further, given a data set which looks like light blue, dark blue, green, and purple, the loss function should be low. That means the loss for blue and dark blue, the loss function should be low, whereas for the blue and green, the loss function should be high. So for example, we can see the similarity of blue and dark blue are much higher than the similarity of blue and green. So what we want is then image I and its augmented versions, like flipped, cropped image, or the or with various illumination differences, both of these should have same representation, whereas an image of tree and image of cat should have different representation. So we push all the representation for similar images together, and we push all the images for unsimilar images far away from each other. So how do we know which are similar and unsimilar images? So you can consider in self-supervised learning, every image is its own class. So given an image, once we do the augmentation, all the image and its augmentation are positive class, and all the images and other images are negative class. So the second type of self-supervised learning method is clustering. So in this method, we try to use a neural network to directly predict the cluster of the input image. So imagine we know that our data set has 3 clusters. So each of them are represented by something called prototype vectors. The goal of the deep learning model is to directly predict which cluster each image belongs to. So given an image and its augmented version, we directly try to predict the cluster assignment of one image from the augmented image. So this can help in removing the negative image bottleneck. So using only a single image and its augmented versions, we can pre-train a model and use it for different application. In the end, at the end of clustering, the features will be grouped together as shown in the right side. All the blue images are in the one block, all the green and all the purple. So the third method is distillation. So bootstrap your own latent is a distillation-based architecture. As you can see, there is a student and a teacher. So the goal of student is to mimic the predictions of the teacher. So given a teacher, which is an average model, we try to distill the knowledge of the teacher into the student network. And then for here, given an input image, teacher and student both make prediction. But the student's goal is to mimic what the predictions of the teachers are at the end of the training. So, and we also use some asymmetry in the architecture and the learning rule to avoid collapse in during training. This will make sure that during training, it's stable and the loss functions don't collapse to infinite or something. So this is another way of learning an unsupervised representation from unlabeled data. So the fourth type is information maximization or Barlow twins. This has two loss functions, invariance and information maximization. If in invariance, we want image and its augmented versions to have same representation. Whereas for information maximization, given a representation, each dimension of the representation should learn unique concept. For example, one neuron here learns a concept of dog and another neuron here learns the concept of cat on the right side image. So maximizing the information content helps us to learn efficient representation. Same as humans, we don't want to use all our neurons to learn same concepts. We want to use different section of neurons efficiently by learning different concepts. So the data set today that I'll be using is soybean stress classification data set. This data set was collected by Professor Arthi Singh's group at Iowa State. In total, we had 16,500 images approximately, and we have 9 classes. 8 were infected and 1 were healthy. And the classes were bacterial blight, brown spot, frog eye leaf spot, herbicide injury, iron deficiency chlorosis, potassium deficiency, bacterial pustule, and sudden death syndrome. So as you can see, the data is imbalanced. The healthy class had 4,000 images and the infected class around 1,500 to 2,000 images. So to summarize again, we talked about 4 different ways of learning representations from unlabeled data. One was contrastive, clustering, distillation, and information maximization, which is called Barlow twins. He's a neuroscientist, that's why the name Barlow. And so all these methods use different kind of loss functions to learn the representation from unlabeled data. In contrastive and clustering and distillation, we try to maximize the similarity between same images and try to reduce the similarity between the different images or different kind of images. In Barlow twins, we try to have 2 losses. One is invariance and another is information maximization. Both will help us to learn efficient representations for the encoder, which can be used for the downstream task. So this is the important slide. If there is one takeaway from this talk, this is the key results. So given 14,000 images for pre-training, we train a model using 14,900 images for the pre-training stage. That means given a set of unlabeled data of around 14,900, we use all the 4 contrastive losses or the self-supervised learning loss, which we spoke about earlier, to train this encoder model, which is ResNet 18. Once we finish training, the ResNet 18 model gives a feature representation of dimension 512. Now, we are going to use this feature representation of dimension 512 for training a linear classifier on the few amount of labeled data. So we get this representation from unlabeled data and use the representation for some downstream task using labeled data. For example, if you use 3% of training data as labels and then fine-tune our representation, we get 91% accuracy using Barlow twins, which means 3% of data is 450 images approximately. So just 450 images we are labeling out of 16,000 images. Labeling just 450 images out of 16,000 images can give 91% accuracy for deep learning model is a big thing, because generally we assume deep learning models need lot of training data and lot of labels, manual annotation. But here, because of self-supervised learning, we can drastically reduce the amount of label data needed. We can see that almost all the self-supervised learning methods perform really well. For example, for 10% of label data, all the methods get classification accuracy of 90%, more than 90%. And Barlow was the best performing method, which indicates contrastive performed, information maximization method performed better than contrastive, clustering, and distillation. So the keynote is in future, we'll collect more amount of unlabeled data and then use it for training encoder model. And then once we have this pre-trained model, we will use the pre-trained model for our downstream task using transfer learning. So for example, the representation, we can use it for identifying the nearest neighbors. The first image was the input image, and all the other images are the nearest neighbors for the query image, which is shown on the left-hand side. The two images show that all the similar images are clustered together in the representation space, as I explained earlier, using color images. So, but there are some challenges. How to use data set from another domain? Like we have disease data sets collected by multiple different groups. For example, Penn State has publicly provided a plant village data set, which is around 45,000 images. Can we combine soybeans with plant village data set to get a better pre-training model and then use it for downstream application for transfer learning? We tried that, but unfortunately, no, we are not able to use efficiently out of domain data sets from other crop species. That's an open challenge. So although we tried to use a large amount of pre-training data, the quality was important than the quantity. So data quality and how relevant they are to our task is really important for the pre-training stage. So the second problem in using self-supervised learning is we try to combine active learning and self-supervised learning methods. So I think Aarti Singh spoke about active learning methods where we adaptively sample the data samples for training or deep learning model. But we see that pre-training using self-supervised learning, using large amount of unlabeled data, and then using active learning on top of it provided no gain compared to just regular randomly selecting samples for doing the transfer learning. So the key point is active learning was not helpful when we are using self-supervised learning methods, and we are still working on how to efficiently make active learning work with self-supervised learning. So here you are seeing a UMAP visualization of the feature vector, which we got from self-supervised learning for all the class. This two-dimension visualization is got in unsupervised manner, like similar to PCA, but we use a deep learning model, got a representation, and then we reduced it into 2 dimensions and visualizing here. And you can nicely see different classes were clustered together after applying contrast learning method, which is simpler. So on the right, we are seeing like we can sample the data from this in 2 different ways. One is random, another is submodular. Submodular means you are sampling diversely, whereas random might sample multiple images from the same cluster, and the performance might go down. But submodular selection was not helpful in our case. In conclusion, we can see that we can remove the need of labeled data needed for pre-training using self-supervised learning methods, and it can help in data-centric AI. That means we can identify the best data set for a specific model to achieve maximum performance. Till now, given a data set, people used to try different models to get maximum performance. But now we decide that I want to get the best performance for ResNet 50 model. What should be my data set? So we are going to curate our data set to get best performance for our deep learning model, not the other way around. It's not that we have a fixed data and we are going to train different models. We are going to fix the model and identify the best data sets. That's the future. So in future, everyone will be working on identifying how to collect data, how to annotate data, and how to clean data. And self-supervised learning methods can really help in doing this process. So these representations can also be used for image retrieval and removing outliers. As I told, the first step in training a deep learning model is cleaning the data. So for cleaning the data, we can use the representations and then use image retrieval algorithm to group all the clean images and the unclean images separately, and then train our model. And it provides 2 benefits. One is model initialization, another is diverse sample selection. So we can see that even though we have just unlabeled data, self-supervised learning methods provide a lot of benefits for transfer learning. And in future, I believe that no one will use labeled data for transfer learning, and it will be just unlabeled data for transfer learning. So, but for fine-tuning stage, we need like 3 to 5% of labeled data than what we used to do previously, and we can give around 90% accuracy easily for soybean stress classification problem. Thank you.
Great job. Thank you for the nice talk. And at this time, we'll take questions for either speaker. So anyone in the audience have a question?
Can you comment on the so the image that you showed where it was the table with different methods? Can you comment on the amount of time it took for each of those methods to do training and human input versus the amount of time it took each of those methods to run versus the quality of the data you got out of it? So was there an impact for running time versus the amount of human input time to train those models?
We can train these models in 12 hours if we use a lot of GPUs. So I use like 2 to 3 GPUs. So, in deep learning, we need a lot of GPUs for training these models. Human hours, it's just one-time task of collecting unlabeled data, as I told. So for labeling 450 images, you can imagine that if each image takes 1 minute given for a class, you can see that we can get a lot of benefit out of deep learning models compared to humans because of GPUs. So the GPU are the winners.
Any other questions in the room? Okay, have you tried the learning algorithms for full plants, images, and/or in-field crop disease identifications?
I've not worked on in-field disease identification, but I'm using these methods for in-field images for other applications. For example, semantic segmentation. So all these methods I want to point out are not only for classification, it's for object detection, segmentation, and other tasks. And we have also tried these images for full plant images, and it works really good for segmentation, for maize, not for soybeans.
Great. Any more questions for either speaker? Here's one more question from Anjali. For the soybean stress, did you classify into stress versus non-stress, or classify into each of the stresses, i.e. bacterial blight, nutrient deficiency, etc.?
Thanks for the question. Yeah, we classified into 9 classes separately, and we didn't do binary classification. So yeah, if we don't have large amount of unlabeled data for self-supervised learning, we have to initialize the self-supervised learning models using ImageNet data. So before we start our self-supervised learning procedure, we have to start from an initialized pre-trained model, which is from ImageNet data. Generally, in a computer vision community uses a dataset called ImageNet to start training the models. And even for self-supervised learning, we can do the same thing if we want to do it with small amount of data.
John's question. And then for Anjali, regarding the image capturing, did you try different angles, number of pictures, and if, how big of a difference did that make? And do you believe using just 8 pictures over or underestimated the surface area on average?
So we did not test we didn't test the whole analysis, no, based on a different number of angles or pictures. We chose that largely because that was the maximum number of pictures that we could take within an 8-hour day and get our entire population done. So it was more logistics, to be honest, for one person to do it all. It might, I don't know, I can't say for sure whether it would underestimate or overestimate, but it might impact that, and it might depend actually on whether or not the plant is wilting, right? Because as your plant wilts, you're hiding some of the leaves inside the plant. If your plant is larger, you can see everything. So it might impact surface area a little bit.
Great. Any final questions? With that, let's, got one more in the audience here.
Thank you for the great presentation. So my first question goes to Anjali, and it's about, did you consider using a biomass? So like just cut the plants and weigh them and see if you have any correlations and if you get any QTLs. The second question is to Koushik, and I'm impressed that, I found out about the plant village the other day and using that data set. But then the question would be, across species. For example, bacterial wilt is, can be found across species. And, you alluded to the fact that you tried to use the data set from plant village and it didn't work really well. So do you think if you had, say, bacterial wilt images from different other species, your model would be better or at prediction or something like that? Thank you.
Let's let Anjali answer first.
So we did not do any mass because that's destructive, and we could have done that at 8 DPI, but we chose not to. We were just looking across the whole spectrum.
So yeah, what we observed was we should be careful in which data set we are using for pre-training and not just randomly select all the classes. So some classes are hurting the performance than improving the performance. So it makes sense to use classes and diseases which are really helpful for transfer learning and not just random images. And I have one more question for the first speaker. So, hi Anjali. If we, for example, did you face any challenges in semantic segmentation of the plants? Yeah, could you please talk about that?
Yes, actually, that took a really long time. I thought it would be very easy, but no, it wasn't, because you can imagine all the branches are blocking each other. So I was interested in your talk because we went and the students had to label. And so they did develop a very quick, not very quick, but a much quicker way to identify the stem. So we ended up doing a quick stem identification, and then we went with either side. But yes, that was hard. That took quite a while.
Thanks.
Okay, and another question for Anjali in the chat. What type of sensor might you hypothesize would give you the best data to improve your QTL analysis? Could it be multispectral or something totally different?
I wanted to use hyperspectral. So I was thinking we've done some really, really preliminary experiments that show that when other people have done a lot of other experience showing that, yeah, hyperspectral will give you differences between wilted plants and non-wilted plants. So I think that would be one thing to do. I don't know if that would pick up, though, the QTL that was on chromosome 6 that we missed. I'm not confident that would do it, because I think that is more based on what your eye can see visually compared to our 2D images. But I think it would be really cool to see what the hyperspectral picked up. We haven't done anything, though, past those really early experiments.
Have you gone back to visually look at the images of plants that segregate with each, with that interval for susceptibility or resistance?
That's on the list, yeah.
Thank you. Okay, any last question? With that, let's thank both our speakers for 2 engaging talks, and everybody for attending.